Okay, now on to part two. We're going to start with the sliding filament theory. Okay, so what is sliding filament theory? Um, First, it just explains how muscles, proteins interact and produce force. But let's kind of go back to 1954. So, muscle contraction remained a mystery until an investigating theory was proposed by two groups of different scientists in the mid 20th century. The sliding filament theory was proposed in two scientific papers not until 1954 in the Journal of Nature, which is a very prestigious journal. These articles provided experimental evidence revealing how the muscle shortens and develops force. This is the most insightful explanation of how muscle proteins interact and produce force and cause movement of the muscles. So we know from the last video that the filaments will slide over each other to produce force without changing in length. So kind of know that is actin and myosin, they don't change in length themselves. The sarcomere does change in length. So when they slide past each other, the sarcomere will shorten, but those filaments remain the same size. The sliding, though, that's what produces the change in the striation pattern that we learned about earlier. Something else is the number of actomyosin complexes formed determines how much force is produced. So if we have, let's say, 50% of our actin bound to myosin, then we're only going to be able to produce about half the force that our muscle is able to, compared to if we have almost all or every single actomyosin complex formed in the muscle, then we're going to be able to produce maximal force. So yeah, all of force we produce is determined by how much cross-bridging or actomyosin complexes are formed. So of the sliding filament theory, let's first start with this resting phase right here. So at rest, we can see the actin and myosin are not touching. This picture shows they're not touching at all, but some models have described that actin and myosin may still interact, but in a very weak bond, because tropomyosin is covering most all of the active sites on actin. But there may be some active sites that are still available, and the myosin head right here can attach onto it. So if they're touching during rest, it's a very weak bond. So cross-bridge of myosin is in close proximity, if not touching actin, but they're not interacting all the way because those active sites are blocked by tropomyosin. You can see in this picture by the blue strands. This is called the steric blocking method. The essential displacement of tropomyosin is triggered by an increase in muscle fiber systolic calcium concentration. So on the next slide, we'll learn about calcium, but you can see it coming in. We see right here, these calcium molecules are coming and they're going to bind onto troponin, which we learned that troponin C binds onto calcium. That's going to be the stimulus for the shifting of tropomyosin off of the active sites so actin and myosin can bind together. So resting phase. Very little calcium in the myofibril. It's stored in a structure called the sarcoplasmic reticulum. We'll see a picture of that reticulum later. So that's the resting phase. There's not much calcium. Excitation, contraction, coupling phase is next. So this is initiated at the synapse of the muscle fiber. The synapse is like the connection point. And we'll learn about that more in chapter 5, but just know it's initiated at the synapse. The electrical impulse, that's our action potential. So our action potential, our electrical impulse is generated, and it's going to spread across the muscle membrane, which is the sarcolemma, which we know exists just below the endomysium, the connective tissue layer. The sarcolemma has a really nice environment, we said, of sodium, potassium, and calcium, and that allows the action potential to travel across the sarcolemma. What the action potential is going to come across is something called a transverse tubule. This is like a little invagination or a little tunnel inside the muscle. So it's a little tunnel, and the action potential is going to slide into. It's going to propagate down into that action potential. And that's when it's going to penetrate the sarcoplasmic reticulum. This is where we house all of our calcium. So there are some receptors that have to be activated first in order for calcium to be released. So the DHP receptor is first. The impulse travels down the T tubules and hits this DHP receptor, which acts as a voltage sensor. This is then going to interact with the ryanodine receptors, which are located in the membrane of the sarcoplasmic reticulum. And they're going to open once stimulated, and that's going to cause the release of calcium into the systole of the cell. Calcium will then bind to troponin C, which we talked about earlier, and that's going to cause the shift in tropomyosin off of the actin binding sites. Now, myosin has the opportunity for those heads to swivel and interact with those active sites. And once they can attach to the actin, that forms the cross bridge. And then it's going to pull actin towards the center of the sarcomere. That's going to shorten the sarcomere, which is going to shorten the muscle, and that's going to cause force. That's called the power stroke, that phase right there. This movement's described as a ratchet movement, where that myosin head will swivel at a hinge pivot and pull the actin filament over the myosin, and the sarcomere shortens. And we'll learn more about this step in just a second, but let's look at a picture of this. So here's this whole thing is a muscle fiber. This right here is the sarcolemma, which is underneath the endomysium. And then these guys right here are the transverse tubules, those little invaginations in the muscle. This here is the DHP receptor where it's located, along with a ryanodine receptor. So they're located in these membranes. They act as a voltage gate, so once they're activated, they're going to open and release calcium from the sarcoplasmic reticulum. So the reticulum is all the blue. And so once they're activated, calcium will then release from this blue structure here, and it's going to flood into the myofibril where actin and myosin exist. So here's this picture kind of shown a different way. So don't worry about step 1. We'll talk about that in chapter 5. But let's talk about this action potential. It's going to propagate into the T tubule. So here the blue is sarcoplasmic reticulum. Here's calcium. Calcium is then going to release from those little channels. So these channels for calcium open. And that allows the action potential to kind of continue to travel through the muscle and activate more sarcoplasmic reticulums. And as calcium is released, we can see it will then bind to troponin. So it's going to bind to troponin right here and cause the shift in tropomyosin. And now this head right here can attach to those actin sites. And it's going to swivel. If you follow this arrow, that head's going to swivel back and forth, and it's going to shorten actin. So myosin attaches to actin, releases energy stored from the ATP hydrolysis, and moves that head. This phase, step 5, is called the power stroke. We're about to go into step 5, the power stroke, in a little bit more detail. Once we're done with the contraction phase, contraction terminates when the calcium is pumped back into the sarcoplasmic reticulum. So this calcium here, it doesn't get wasted. It doesn't just like disappear. It's pumped back up. So we have this ATP pump, and that pump is going to help get calcium back into the reticulum. But let's go into step 5 in more detail. So we see here that this just shows one head of the myosin. Just know there are lots of heads that are going to go through the same motion at the same time. So the myosin head will swivel at that pivot that we talked about. And we see here that it's energized. What does it mean if it's energized? That it's using ATP to do this. So we have to have ATP present in order for this process to occur. So the energized myosin head will attach onto it, and then we see here, once it's bound, it will then pull. It'll then pivot. So it'll pivot, and that's going to shorten. So the movement you see here is to the right. It's going to pull that actin towards the M line. The contraction phase. This is that cross-bridge flexion. Flexion, we know, is the pivot part. It's caused by ATP breakdown. Think back, we said that the myosin head is made up of an enzyme, myosin ATPase, and every time an ATP attaches onto that head, it hydrolyzes. It breaks apart into ADP and phosphate, and that phosphate is used, and that's the energy. The second ATP that comes in is going to replace the ADP, and this process will just occur over and over again. So that myosin head will detach, and then it will recock, and then it will reattach to actin. If you're confused, I promise we'll show a video and a picture of this in a second. And then relaxation occurs, so we have no more movement. Calcium is pumped back up into the SR, and then actin and myosin return to their unbound state. So I clicked on that link at the bottom of the screen, and it came up with this animation. So this just kind of shows you how that myosin head works. So the myosin head just doesn't pull once. It's constantly pulling and shortening, because you can see it doesn't move very far when it pulls it. So this is the pivot, and this is attaching to actin. If you look at the blue, that's the tropomyosin. You can see it shifting off the actin every time the myosin head moves towards it. What all is going on here? Okay, so the yellow, do you see the yellow ball flying at the pink ball there? That yellow is supposed to depict calcium, because as calcium attaches to troponin C, that's causing the shift of the tropomyosin. And then we see up here this green guy and this triangle. That's going to be ADP or ATP that energizes the head, and then it breaks it apart and it turns into ADP. So let's talk about this process a little bit more. Okay, we read that statement at the top there, and we already learned that the number of cross bridges that are formed, the number of myosin heads that attach to actin, that determines how much force we produce. But this picture here is really important because it shows that power stroke one more time. So the myosin head, we see here a high energy configuration. So the cross bridge attaches to the actin filament. We needed ATP to do that, and what happens is now it's hydrolyzed into ADP and phosphate. All right, so it attached, and now we see here's the working stroke. So here's that pivot. So now ADP and phosphate are released, so they break off. The myosin head pivots and bends as it pulls actin towards the middle, so sliding towards the M line. And then look over here. Here comes an ATP. So another ATP molecule comes in, and that's what causes the release. So a new ATP attaches to that head, and now the myosin head, or the cross bridge, can detach. It can break off of the actin. So we needed that ATP, but oh my gosh, if the ATP attached, what happens? Look up here, ATP hydrolysis. So it's going to break apart again, but it's energized. So now that energized head, the phosphate provides this energy, and it can attach back up, and the process can happen all over again. So that's the power stroke phase.
Or relax when they receive terminal, and sarcomeres run adjacent to one another down the length of the myofibril appearance. The muscle contracts the M line, both sides when active the myosin, although the edges of the sarcomyosin. The thick.
Oh my gosh, so sorry that you have been watching the whole time. I was trying to get to the point that I actually wanted you to see. So sorry you had to watch that. But now this is the important part, and this will kind of wrap up the sliding filament theory.
Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract.
All right, so we'll stop it there. I hope you understood kind of the sliding filament theory and kind of everything that kind of goes with that. Let's move on to proprioception. So this is another question on your assignment. First, what is proprioception? It's basically how the body senses where it is in space and the sense of our neighboring body parts. For optimal performance, a constant flow of information about the body's position needs to occur in the neuromuscular system. This is basically called a constant stream of neural feedback. It's also called our kinesthetic sense. It's the brain's integration of proprioceptive inputs via receptors located in the muscles and the tendons. It's information sent to the conscious and subconscious portions of the brain. This allows for a repeated complex skill to just feel right. And that's what the learning effect is. So we have all these proprioceptors located in our body, but the ones we're mainly going to focus on are GTO and muscle spindle. So we'll learn about each one of those on the next slide. But what are these kinesthetic receptors? What do they do for us? So like I said, it allows for a repeated complex skill to just feel right over time after practice. It does require successful, perfect practice to learn a motor pattern to reproduce it over and over again. But it's conducted so quickly that we don't even have to think about it after we get really good at it. And that's what the learning effect is. So it's important, especially when we learn a new skill, because it keeps the central nervous system constantly informed as the body is learning the new movement subconsciously. So important just anytime we do something new or trying to get or become perfect at a skill. Okay. Muscle spindles. So muscle spindles are gamma motor neurons. These are lower motor neurons that take part in the process of muscle contraction. They don't directly adjust the length of the muscles like the alpha motor neurons do. Their role is important though in keeping muscle spindles taut and healthy, allowing for continued firing of the alpha motor neurons, which leads to muscle contraction. So the main role of the muscle spindle is to monitor length of stretch and rate of stretch. So how much is the muscle stretching and how quickly did that muscle stretch? And then what its innate response is, it's to initiate a contraction. So if we see here, if we follow the steps, the stretching of a muscle spindle is stimulated. First, I kind of forgot to mention muscle spindles, they're interphysial fibers. So, interphysial meaning they're embedded inside the muscle, and they consist of different types of fibers. There's one dynamic nuclear bag fiber, one static nuclear bag fiber, and 5 nuclear chain fibers, all composed of the stretch sensitive tissue. Um, and so, what this tissue will do is it will detect the stretch, and then it activates a sensory neuron, which is sent to the spinal cord. And it's processed at the spinal cord, and then a motor neuron is sent back to the same muscle that was stretched, and then that muscle contracts. So, my question for y'all is, why does the muscle contract? What's the point of this response? It's a protective mechanism. It prevents the muscle from being stretched too far. So again, I would have you probably stand up right now and go down and touch your hamstrings, but touch your hamstrings, touch your toes, go and touch your toes really quickly, and you're gonna feel your hamstrings fire, and you're gonna feel them contract. That's also the muscle spindle. That's called the stretch reflex. So the stretch reflex is when a muscle is quickly stretched, and that initiates an immediate contraction. So a fast stretch, immediate contraction, and what happens here? It activates titin. We just got done learning about titin. So it activates titin during that reflex, which acts as that molecular spring, and it limits range of motion. So that's our muscle spindle. These spindles are active in all sports and exercise that involve any degree of muscle elongation, especially muscle elongation at a rapid pace. The stretch reflex occurs basically to prevent the muscle from tearing, and it contracts that muscle so it shortens it rather than stretches it further. Next is GTO. So GTO, main function, well first let me start back. GTOs are located in the tendon, not the muscle, and they're extraphysial fibers. Extraphysial fibers located in the tendon near the myotendinous junction, so in between the muscle and the bone. The GTO functions to protect the muscle and the tendon by responding to tension. So it detects how much tension is placed on the body. And the GTO will fire when there's a heavy load placed on the body. And then what's the main role here? To relax the muscle. So the role is to detect tension, so excessive force placed on the body, and instead of contracting, now the body starts to relax. Those muscles relax, the tendon, everything kind of shuts down. This is an innate response. This is how our body was designed. But let's think about resistance training. When we lift weights, let's think about back squat. We've got a heavy barbell on our back, and that heavy barbell is causing a lot of tension on our tendons. And when we go down in the bottom of the back squat, if we're at the very bottom and the tendon and the GTOs start to fire, if we let the GTOs do its job, what would happen? The muscles would relax and we would just fall to the floor. But with resistance training seeks to inhibit or stop the action of the GTO. So we can actually override the GTO, which allows for greater force production. Because we don't want to relax when we've got a heavy weight placed on our body. We want to contract the muscle to produce force. So that's overriding or inhibiting the action of the GTO. GTO inhibition is what it's called, and that allows for greater force production. So pretty neat. Now let's answer this question here. How does heavy exercise affect our proprioceptive sense? So heavy exercise meaning we just did a really hard workout. It was really intense, it was really hard, and it was a really long workout. What happens at the very end of that workout? How are we going to feel? How's our body feel? It's going to feel tired, right? It's going to feel fatigued. So our proprioceptive sense of our body's position, it's affected by fatigue. The more fatigued we are, the decrease in our proprioceptive sense starts to occur. So the importance of training and conditioning and becoming more trained over time is to reduce fatigue during exercise. The goal is to enhance the flow of information from the muscles to the central nervous system and back. So my point here is when we're fatigued after a heavy workout, our GTOs and our muscle spindles are less responsive. They don't work as well. So if they don't work as well, then our central nervous system is not getting the information that it needs. So we're affected by fatigue. So in order to not be affected by fatigue, we need to become more trained. And the more trained we become, our body learns how to handle fatigue and still have appropriate communication with the central nervous system. So yeah, pretty interesting. All right, let's switch to the last topic of part 2, and that is fiber type. So with fiber type, we first want to learn different classification systems of muscle. Okay, we have slow twitch and we have fast twitch. We also have type 1, type 2A, and type 2X. And then we also have SO, FOG, and FG. So what does all of that actually mean? Obviously, twitch speed, so slow twitch speed versus fast twitch speed. The type is just how we refer to them. And then this here, we can talk about this. SO means slow oxidative, meaning it uses oxidative metabolism for energy. F is fast for both of these. O is the oxidative, and then G is glycolytic for both those G's there. And we'll learn more about that in just a second. But skeletal muscle basically possesses several different muscle fiber types with various metabolic and then performance capabilities. These different classifications are developed on the basis of the different histochemical and biomechanical and physical characteristics. Fiber type is genetically predetermined. So no matter what fiber type we want to possess, it's based on genetics. So it's established shortly after birth. So you'll get kind of a mix of what your parents had. But also, skeletal muscle is just a big mixture of all these different types throughout the body, and you can see that. Let's talk more about the classification systems. We already went over type. So we have type 1, type 2A, and type 2X. Speed, that has to do with twitch speed. So how fast do the fibers twitch? Slow versus fast. Metabolism, we just went over that as well. Do they use oxidative or glycolytic, or a combination of oxidative and glycolytic? Color. So we have red versus white fibers. And don't look at this picture because it shows type 1 being white, but this is just the stain. These fibers are stained with a different chemical. But if you were just to look at muscle itself, type 1 fibers are going to be more red in color, and then type 2 fibers are going to be more white in color. The red is due to the myoglobin. So the red color, more myoglobin, more oxygen, more blood flow, versus the white fibers, which don't have as much myoglobin or blood flow. But yeah, that's kind of our classification system, and we'll kind of continue to talk more about this. One thing to know, though, is that subtle changes do take place with our fiber types over the lifespan, and it depends on our activity level, the type of exercise we do, hormonal concentrations, aging related changes, and then disease. All of that will affect changes to our fiber types. Okay, let's talk about type 1 slow twitch. What are the characteristics? And remember, fiber types are this is the last question on your assignment. So this is number 5, the critical thinking question. Okay, so slow to reach peak force and low peak force. We see in the picture down here, we're looking at the red. So the red is our slow twitch, over here is force over time. You can see that it doesn't take very long or we can't reach very much force at all, and it takes a little bit of time to get there too. So that's the first bullet point. It's slow to reach its peak force. And when it finally does reach peak force, that peak force is very low. But the benefit of this fiber is its high oxidative metabolism. So it uses aerobic metabolism, it uses oxygen to produce energy. And it's fatigue resistant. So based on those characteristics, what activities do we rely on type 1 fibers? Your guess is probably correct. Aerobic endurance events. So low force activities that require that are kind of fatiguing over time. So like running a marathon or any type of running, like slow distance, long distance running. That's going to use type 1, where we don't want to fatigue out quickly, where we want to maintain our energy for hours possibly, but we're not producing high amounts of force, and we're not producing force very quickly. So like walking, jogging, cycling, long distance swimming, anything kind of long distance related. So I'm going to give you some other characteristics of type 1 fibers. Greater capillary density. So basically these fibers have more capillaries, meaning we get more oxygen and nutrients due to a greater blood flow. Higher myoglobin content, again with the greater oxygen, receives a rich blood supply and oxygen. It also has really good mitochondrial density, so more mitochondria, which we already know about mitochondria, produces ATP. Fatigue resistant, so contraction over long periods of time at low intensity without getting tired. And then it has a really high amount of oxidative enzymes. What about fast twitch fibers? So we have 2 different types of type 2s. We have an X and an A. So the A is your intermediate fiber, and we'll talk more about that in a second when we get down here. But X is an intermediate, and that's your FOG fiber. Or sorry, A is your intermediate, and that's your FOG. It uses both oxidative and glycolytic. The X only uses glycolysis. Okay, so what are some characteristics here? Rapidly develops force. You see in this picture here, when a fast twitch fiber contracts, it is fast to reach peak force, and it produces a high amount of peak force. But it has very low oxidative or capacity for oxidative metabolism, but high capacity for glycolytic metabolism. It does fatigue out very easily, but it's good for that's your question. What activities do we use type 2? Type 2s are used for high intensity, but really short duration activities, like a short sprint. So sprints or resistance training, where we're producing lots of force. A lot of sports with really fast movements rely on type 2s. So anything that produces a lot of force and really fast contractions. Other characteristics, they produce ATP rapidly, so very fast, but in a low capacity. So we already know low aerobic ability, but high anaerobic ability. They lose force quickly, but they produce a lot of it in that short term. They have fast contraction speeds, very high glycolytic enzymes, and they have more protein content. These fibers also grow more easily, meaning hypertrophy. They're going to experience larger growth, compared to the type 1, which does not grow as much and doesn't produce or see as much hypertrophy. Let's look at this picture down here. So this is our type 2X, and this is showing a shift in muscle fiber types. You can see that we actually do have more than just 3 fibers. We have multiple subtypes, but the main fibers are the X, the A, and the 1. So what is this picture showing? This is from a research study that showed that type 2X fibers are actually very inefficient because they fatigue out way too easily. And that the more trained we become, and it doesn't matter what type of exercise we do, if we lift weights, if we run, if we cycle, if we do yoga, it just matters if we have muscle contraction. So if the muscles are contracting, we're going to see a shift in the X to the type 2A. You think, well, why would we want to shift? And I even kind of show it here. We've got our X and we shift to the A. This is actually a very beneficial change in fiber subtypes. It means that this type 2A or this type 2A can produce a lot of force, but it doesn't fatigue out as quickly as the type 2X does because it has more myoglobin and more mitochondria, so it's a little bit more oxidative. So this is a beneficial shift here, and that's kind of what this picture shows. One thing to know is that type X and A, they can switch back and forth depending on what our activity level. If we become sedentary or inactive, they're going to shift back to the X. And if we become very active and start exercising, we're going to see the shift to the A. But one thing to know is a type 2 can never become a type 1, and a type 1 can never become a type 2. We see a question mark here. It just means research has not shown that yet. Okay, this picture here, or this chart here, is a really great chart to study. It goes over those characteristics that I just discussed, but it just separates them in type 1 and type 2. So just think of the X and the A are both here in this type 2 category. Most important things we see here are going to be ATP stores and phosphocreatine stores. We learned about this in chapter 2. So we can see here low versus high. Glycogen and triglyceride stores. We probably talked a little bit about this in chapter 2 and 3, but this talks about type 1s have a higher fat content, wherein type 2s have a lower fat content, but a higher protein content. Glycogen stores are very similar though. Capillary density, mitochondrial density, you can see the difference there. But yeah, this is a great chart for studying. You do have to know all these characteristics. Let's talk about how do we determine fiber type. So how is fiber type determined? Best way to determine is by a muscle biopsy, where you cut a thin cross-sectional slice of the muscle and then you would stain it. And then this is the staining procedure. Let's talk about how would you do this. So we see here we have 3 different pH conditions or pH solutions. So we have 2 acid baths and an alkaline bath. This histochemical staining method will result in a different stain intensities, which you see here among the fiber types. And they're divided into categories and assigned to either type 1, type 2, X, A, C, kind of all of that. And usually they're all unique shades of gray is what you would kind of see here. But yeah, we have different pH conditions down here. And then we look kind of at the X, the 2A, and the 1 is what we're concerned with. And you see here the 1 is going to respond to this acid bath, so with that lower pH level. And then both the 2A and 2X we see are going to respond to the higher pH level, that pH of 10, that alkaline bath. So with those different pHs, again, it's just a different solution. I don't really get into this. I don't do research in this area, but it's kind of interesting. It's just a different staining procedures. The staining procedure I'm familiar with stains with the enzyme myosin ATPase, which we just learned about. Myosin ATPase has to do with contraction speed. It has different isoforms where some result in quicker enzymatic reactions for ATP hydrolysis than others do, thus higher cycling rates for the fibers in that actomyosin interaction. So myosin ATPase is directly related to speed at which the myosin head will bind to actin and swivel to generate force. And it provides functional classification based on the muscle fiber shortening velocity. So the greater oxidative capacity, we're going to have more type 1s. The less oxidative is going to be more type 2s. But you don't have to know all those crazy details. You just have to know, no, you don't even have to know anything. You just have to know that we determine fiber type by a staining procedure with muscle biopsy using myosin ATPase as the enzyme to stain the fiber types. All right, here is the myosin molecule. So this also helps to determine fiber type. We can look at here different myosin heavy chain content. So first let's kind of define all of this. What are we even looking at? So this is our myosin head. This is our tail, which is a heavy chain. So this MCH, that's our heavy chain, and that's what we're looking at here. We have up here the ELC, essential light chain. It's the inside of the head. And then the regulatory light chain is this RLC. And then also heavy chains are the cross-bridge head. So the head of the myosin we know is made of 2 heavy chains and 2 pairs of light chains. We can use the myosin heavy chain composition to determine fiber type. This allows for greater understanding of fiber type adaptations that occur with exercise training, and also helps us understand the transition of type 2X to type 2A. So if we look here, myosin heavy chains, we see the type 2X and the type 2A. What do you notice? It looks very similar, and they're grouped together, where myosin heavy chain for the type 1 fiber, is not grouped with the type 2s. This shows why a type 1 fiber cannot become a type 2 fiber. It's due to differences in the myosin heavy chain composition, whereas a type 2A and a type 2X can transition back and forth based on the physical activity and inactivity levels of a person, because the heavy chain content is so similar between the X and the A. But the 1 cannot become a type 2. So yeah, we are finished with part 1. You can now complete your assignment. I really think we have covered everything. So good luck with your assignment, and glad you watched part 2. The next video will be over part 3.